Hello, we are going to start Module 4, the Immune System Lecture Part 2. In this lecture, we're covering Section 7.3. I would say this is the one of the longer of the lectures, and it's got a lot of material, so you'll definitely need to do your own notes using the guided notes. We're going to be covering the adaptive immune response, B cells, antigens versus antibodies, and primary versus second response. So at the end, you should understand what all of these are. Last lecture, we talked about the innate immune system, which was that part of the immune system that's always there. And we discussed the inflammatory response that included the macrophages, neutrophils, and monocytes. Now we're going to discuss the other branch of your immune system, which is the adaptive immune system. All right, so don't think that it's one or the other. When the adaptive immune system is occurring, the innate is still working, it's still always there. All right, so the adaptive immune system is activated in response to what we call an antigen. So that's why I say adaptive immunity triggered by antigens. And so here I have a picture. I have a picture of a bacterium and a virus, and these pictures are supposed to help you understand what antigens are. So antigens are molecules on the surface of pathogens. For our class, pathogens are bacteria and viruses. These are organisms that can cause disease, and these organisms have membranes and cell walls, right? And as part of those components, there's specific proteins to those organisms. So here I have little squiggly that's supposed to represent a protein or carbohydrate on the surface of this bacterium. And this molecule, this component of the organism, can function as an antigen. So an antigen is a surface molecule or a molecule that your immune system recognizes as foreign and triggers your immune system to start working very specifically to fight it off, this particular organism. Viruses also have surface proteins. Here I've drawn a cartoon picture of a coronavirus since we are currently on lockdown. And there are specific proteins on the surface of, let's say, a coronavirus, which will trigger your immune system to start fighting it. So we say antigens tag the immune system, right? They're saying this is not a human cell. Antigens give your immune system like a ID code. Like, this isn't part of your cells. This is foreign. Let's attack it. So that's why they tag it and distinguish self from non-self. Whereas in the innate immune system, we talked about macrophages, neutrophils, monocytes. For the adaptive immune system, we're going to categorize these white blood cells as lymphocytes. So lymphocytes are white blood cells that are part of the adaptive immune system. The two types of lymphocytes that are part of the adaptive immune system are B cells and T cells. In this class, we will not be covering T cells, so don't get distracted by T cells in your reading. Use your guided notes that should only point you to focus on the material that is discussed with B cells. So we will only be discussing B cells. So here's a picture, not from your book, of a B cell. It's labeled as B lymphocyte. B cell, B lymphocyte are the same things. On the surface of this white blood cell, there are these little green things, right? These are little receptors, and your body makes all different types of B cells, thousands and thousands of different types, and they differ from each other by the shape of this green thing on their surface. Okay? And we call those green things B cell receptors. And the different shapes allow them to bind to different types of antigens. In this picture, I've found one that looks like a coronavirus, But a coronavirus has a different antigen on the surface than, let's say, the flu virus, the influenza virus. So these B lymphocytes that your body has thousands of, only one or maybe one or two will bind to different antigens on different pathogens like viruses and bacteria that infect your body. Once the receptor interacts with that antigen, right? So once this happens, in this picture, this B lymphocyte is interacting with the antigens on a specific type of virus. Once that interaction, it's like a lock and key, once that is triggered, it's like, yay, we found the right lymphocyte to attack this virus. Make more, make more. And these B lymphocytes start reproducing, and they release something from them that look just like this green thing. And that is what we refer to as antibodies. So they release them, and the antibodies are now not attached to a cell, they're just flowing, and these antibodies can trap all the viruses that are moving around in your body. So we're going to expand on this in a little more detail now. So what I briefly kind of described is referred to as B cell clonal selection. So think of the name B cell, so we're going to be talking about B cells or B lymphocytes, and a specific B cell is going to be selected out of the thousands that your body makes, and then we're going to make many clones of that one. All right, so this picture is not in your book, but I think it's very helpful in understanding this process. You have one that's similar, but I feel like this one's a little more clear. So let's start at the top. Those yellow circles that you see represent antigens. So recall, antigens are specific molecules on the surface of viruses and bacteria. Now here they're just showing you the antigen. They're not showing you the rest of the organism. So these antigens are flowing through your body. And the cells here represent the B cells. And there's many different types of B cells. I told you your body makes thousands of different ones, and they differ from each other by the receptors. So here the receptors are in different colors, indicating that they're different shapes. They're the B cell. One of these B cells will have the receptor, and I've highlighted it here, that will interact and bind correctly with the particular antigen. That's the selection. This B cell has been selected because the pieces fit. Now we're going to make a clone of this B cell. We want more of this particular one. So that's when I say B cells proliferate, which means just start dividing. We're making many more of that one. And in particular, we end up making two different types of B cells from this one. One is referred to as a plasma cell, which is the one without anything on the surface, and the other one is referred to as a memory cell. Plasma cells, the ones without anything on the surface, are the antibody producing cells. So you see here there's an arrow pointing to these structures. So these are antibodies. And antibodies have the same shape as the B cell receptor. Therefore, once these plasma cells are made, they produce the antibody, release it into your bloodstream, and the antibodies start trapping all of the pathogens that have the antigen on their surface that can interact with it. So they trap them, and so then those bacteria can't produce the toxins that make you sick, the viruses can't infect your cells, and they stay trapped there until a white blood cell, a phagocytic white blood cell, will come and engulf and eat it. Remember those macrophages, those white blood cells of your innate immune system. The memory cells are long-lived, the other cell that's made, and they will stay in your body for years and years and years, okay? And this, what we just covered, is the first time you're exposed to a particular pathogen. But then, if you're ever exposed to that pathogen for a second time, right? Years later, or even a month later, or a week later, you still have those memory cells in your body, in your blood, and those memory cells will be activated by that particular pathogen, all right? And this is what we refer to as the second exposure. And those memory cells produce this response much more quickly the second time, because the first time we had to find the right B cell, activate it, proliferate it, all before we could make the plasma and memory cells. Now those memory cells are there, they're ready to go, they interact with the antigen, they make even more plasma cells than the first time, and they also make more memory cells. Those plasma cells are making tons of antibodies. Look at how many antibodies are there versus during the first exposure. So you don't even feel sick. And we refer to, once you've already been exposed to something, you have immunity, right? You have immunity. Right now with the coronavirus, people that have had it have immunity. They can go out and about because they won't get sick again, and then they're not contagious either. So the second exposure is quicker. Why? Because the memory cells are still in the body, which triggers a fast immune response, and you may never feel sick. A little more information on antibodies. So antibodies, remember, are the protein that's released by the plasma cell, has the same shape as the B cell receptor. Here's a more detailed drawing of it. It's a protein, and so these C parts are referred to as the constant regions. Those are the same among all antibodies, right? And the receptors have these C portions too. The V portion is the variable region, and those are the regions that change the antibody shape, which allow it to be interact with different types of antigens. This is a slide I just want to show you so you understand what an antibody actually does. So once the plasma cells release the antibodies and they're flowing through your blood, they do what is referred to as neutralizations. So here the gray structure is a bacteria, but you could also include like a virus in it. The antibodies will interact, lock and key fit with antigens on this bacterium. So they will trap them and they will neutralize them so the bacteria can't release the toxins that make you sick, and they'll get stuck there, and then a phagocytic cell, so this could be like a macrophage or a neutrophil, will come and engulf it and eat them up. Finally, the last thing I want to cover in this lecture is this graph. And this graph is actually just a graphical representation of what we covered in B cell clonal selection 2 slides ago. All right, so let me orient you here. On your Y, you have antibody concentration. On your X, you have time and days. So let's look at like day 1. You have a first exposure to some antigen. Right? Let's say this is the coronavirus. The coronavirus has a protein on its surface, that's antigen X, right? So you're exposed. Well, it takes about 6 days or so for that selection to happen, for the B cell that fits with that protein to be selected. It starts to proliferate, then it makes plasma cells and memory cells, and those plasma cells finally start making antibodies. That's the blue line, level of antibodies. So you're getting up in antibodies, right? So your primary immune response to antigen X, right, under that curve is amount of antibodies. So you have a certain amount of antibodies. Now you're finally feeling better about 15, 17 days after you've been exposed. But during that time, you felt very bad. Then those antibodies have trapped all those viruses, neutralized them. They're not needed anymore, so the antibody concentration goes down. Let's say on day 28, you're exposed to that same virus again. Well, that's why it says second exposure to antigen X. You have those memory cells already in place. So as soon as you're exposed, those memory cells make plasma cells. those plasma cells start pumping out even more antibodies than during the first exposure. So look at the blue line shooting up. So the secondary immune response is after you've already had a response. So you have those memory cells, which are responsible for the more quick response to produce plasma cells and antibodies. So the levels of antibodies are much higher than during the first exposure. So you likely wouldn't even feel sick. And then the antibody levels go down because you are better. And then the red line just means you're probably being exposed to something else, maybe a bacterium at this point or a cold virus. So your immune system is always working.